Hello everyone, it is a great pleasure to be, although only virtual, joining this amazing symposium for the School of Engineering in Osaka. So, first of all, I would like to thank the organizers, in particular Professor Watano, for the kind invitation, and also a dear friend and colleague, Professor Norifumi Kawada. Now, for today, which I hope this is relevant for the School of Engineering, I'm going to present another approach on how we can apply a field of engineering called tissue engineering to actually develop novel therapeutics for unmet medical needs like tissue fibrosis. So these are the disclosure relevant for my lecture, and as you see from the logo, I'm presenting as the CEO of a UCL spin-out company from University College of London called Engitix Therapeutics, and I also still have affiliation as an honorary associate professor at UCL. Now, speaking about engineering of tissue, actually the name of Engitix stands for this, indeed engineering TICS, tissue for developing novel therapeutics. Now, the majority of scientists around the world focus on the cellular environment. We have decided to take a different approach in the last 10 to 11 years by addressing actually the environment beyond the cells, outside the cells. So as you all know, or as you might learn today, this environment is called extracellular matrix and is not simply composed by collagen 1, but actually there are more than 300 proteins that characterize this incredible environment. A lot of people usually associate the ECM to collagen and also to a scarring process and simply therefore as a bystander product of our human tissue. However, we know that the extracellular environment is extremely bioactive and is constantly remodeled to control tissue homeostasis. Now, I think it's also relevant to emphasize that tissue fibrosis and solid tumors are promoted and characterized by changes in the ECM composition and structure. So if we can better understand how these components are affected during disease progression, we can then be in a position to develop, hopefully, novel therapeutics for fibrosis, solid tumors, and aging. Obviously, I'm not going to enter into any level of details here, but just to emphasize that, as just mentioned in my previous slides, the ECM is constantly modulated during different stages of our life, normal and age, but also during disease progression. And there are different players that are affected during these transitional stages. Now, as I said, our biggest passion in research is this extracellular matrix world, and there are multiple applications in the tissue engineering field. So from one side, we can use the ECM for in vitro culture conditions. We can also apply this as a tissue patch to promote wound healing repair, or we can use the tissue desterilization technique for developing ECM that in turn is applicable to preparation of ECM bioink for bioprinter, hydrogel scaffold, cell culture, but also to obtain healthy and disease ECM for understanding how the disease environment at the extracellular level called matrisome is uniquely characterized by set number of proteins that are not expressed in healthy tissues. So Another opportunity is actually to take entire organs that are not transplanted for ischemic reasons and so on, and then to use these as a future structure for a whole organ engineering. Now, from our side, we have been spending significant amount of time in actually developing the sterilization protocols for almost all gastrointestinal tissue, starting from liver. Indeed, we were the first research group publishing how to develop a whole human liver ECM structure, and now we have experience built also outside the GI tissues in multiple tissues and indications. The way as we do this is by applying two different protocols. One is tissue agitation, which is very relevant for small-scale tissues, in particular resection or wedge tissue, and the other is actually for developing whole organ by performing whole organ perfusion. Once the tissue has been decellularized using detergents, enzymes, and tailor the desulfurization protocol according to the tissue of interest, we can then perform multiple readouts like matrixome analysis to identify the unique proteins expressed in this tissue. We can add back the cells and then study the biology of the ECM in modulating cellular phenotype by performing single cell sequencing or RNA sequencing after adding certain cell types like fibroblasts, for instance. We can use the ECM hydrogel for three-dimensional assay and can combine the ECM with a serum from patients to promote the discovery of biomarkers. Now, this is just an example, and feel free to have a look at the publications and send me an email if you would have also additional questions or you're curious on this specific topic. But as mentioned, we have two ways to destroy tissue: agitations or perfusion of whole organ. And what you should take into account, and just remember also for the next part of the presentation, is that after desolidization, the tissue macroscopically turns translucent. And what is essential to show in any tissue, healthy or disease, or from different organs, is that after desolidization, we are able to preserve the ECM structure and architecture, and also we remove nuclei and DNA as a confirmation of acellularity of the tissue. We can do also the whole organ ECM development. This is a liver left lobe, 300-400 grams of human liver left lobe. Similarly, this is an entire all human liver decellularized. Another example is for pancreas or human kidney, and once again, same concept. You can here see with endoxygenous staining the presence of cells after desolidization. Basically, the entire infrastructure of the environment is absolutely preserved without cells. Now, obviously, just to give one example of the approach, starting from obtaining tissue, decellularizing tissue, performing matrixome, applying very fancy techniques like spatial transcriptomic to deconvolute target expression within a spatial context, and then to identify novel targets. One of our main indication of interest is actually primary sclerosing cholangitis. Now, this is part of the liver fibrotic umbrella. It's a rare disease, and unfortunately, there are no therapies for patients affected by this devastating disorder. So, in primary sclerosing cholangitis, patients are characterized by progressive fibrotic disorders, in particular in intrahepatic ducts and extrahepatic ducts. The only treatment is liver transplant, so you can appreciate that this is a massive unmet medical need, and several therapies are currently under development for modulating the bile acid biology or as a direct modulator of inflammation and fibrosis. Now, from our side, we decided to take the discovery of therapeutics for PSC from a different sort of corner. we are very lucky to have access to a significant number of PSC human samples. We have established assay in vitro and in vivo models to confirm or not the hypothesis, and we base our research on two concepts. From one side, to understand the ECM composition of primary sclerosing cholangitis patients, and then to deconvolve how these PSC ECM environment drive fibrosis process forward with the aim of identifying novel therapeutics. So just to put some historical context into what I'm going to explain next, as I said, one of our application or opportunity to identify targets is by studying healthy and disease ECM, and by analyzing the extracellular matrix environment, we enter into the matrisome field. So the actually matrisome word was coined in the 80s by Martin and colleagues when they start dissecting the role of basement membrane in kidney tissue. After that, there was basically a sort of 2 decades gap, if not more, and Naba and colleagues from MIT, they've started to manually curate a list of predicted extracellular proteins using in silico tools, and these represented a tremendous value for the community for appreciating what actually is secreted or is an intracellular protein. However, what we've decided to do is a more unbiased or in silico based approach, but we base our research on tissue ECM matrisome analysis, which allows to have a real representation of the matrisomal environment in healthy and disease status. So first step is, as I said, is to collect human samples, sectioning in a certain way, and characterize that those samples have a clear presence of cirrhosis. You can appreciate with a serous thread, that is a specific staining for collagen, that the liver parenchyma and structure is completely distorted, and you can clearly see how thick these cirrhotic bundles are around the tissue. Now, for non-histological experts, hematoxylinosin staining stain in purple cells, pinkish the ECM, and for the serous thread, you have in yellow the cells and in red collagen. So after visualization, what we would like to see is that the yellow here disappear, and we just retain the red from the collagen. So we have washed out the cells, and this is indeed what happens after visualization. The PSC disorized tissue is indeed characterized by preserved ECM architecture and lack of nuclei, DNA material, and cellular debris. So this is a very good quality ECM, and we can then move to the matrisome study, which in simple words mean to digest those different groups of DCM, extract proteins, barcode those proteins with the TMT tag that allow for semi-quantitative proteomic, run a LC mass spec, and then with our incredible teams of bioinformaticians to analyze the data. So the first step is indeed to perform some quality control on this data by analyzing the distribution of samples in a 3D PCA plot, and we were able to confirm indeed the clear separation of healthy and disease matrisome. So we are happy with the fact that there is something unique or different amongst those two groups. We then perform heat maps analysis to see how those samples cluster as a sort of biological and technical replicates, and it's very impressive to see that there is a clear separation between the deceased and the normal tissue and the biological replicates of different patients' donor cluster amongst each other. So if you recall, one of my first actually message around the ECM is that there are almost 300 proteins into the matrisome. This was something based off of previous funding. We're now able to actually identify more than 300 proteins. As you can see, there are actually 498 proteins that are upregulated in PSC matrisome. The next step is to study the composition of the matrisome by performing a reactor-homogeneity analysis, and we did a comparison with another matrisome just to see whether the in silico prediction was matching our own findings. Obviously, our own findings were addressing a larger set of biology that was not assessed by the in silico prediction. And indeed, we are able to address a significant number of pathways that are ECM relevant, like collagen deposition and ECM reorganization, as well a lot of inflammatory related pathways. So the key message here is that after in the primary sclerosing extracellular matrix environment, the two most dominant pathways are ECM and inflammation. Now, there are still some mysteries on the matrisome, which is around the possibility to understand the spatial distribution of matrisome proteins in a PSC liver. So, you digest the tissue, you have no idea where those proteins actually come from or where they are expressed. Indeed, also, what are the main cellular source of PSC matrisome? So, is only fibroblast, or there is also something more? And then, of course, if we can identify potential therapeutic targets from PSC matrisome and therefore confirming disease matrisome by activity. Obviously, we have, I think, a new world in front of us that we require decades of work to address all mysteries around this ECM environment. Now, in order to address this spatial distribution and cellular source, what, of course, we have decided to spend significant amount of efforts and investment from our teams is indeed the possibility to use an unbiased approach to identify and confirm target expression in the spatial context. As you all know, you can perform immunohistochemistry, and this works absolutely great if you have a good antibody and a validated protocol, but you cannot scale it up for thousands of targets of interest. So in the last couple of years, an incredible techniques has been brought to the scientific community, which is indeed the possibility to perform in a tissue slide spatial transcriptomic, and each of these capture area contains basically 5 millimeter square spots, including multiple barcodes, and the way as it works, in each of these barcodes you will have an oligos with the spatial barcode and the polyase for RNA sequencing. So you can imagine that you load your paraffin embedded section on top of this slide, following some protocols. Basically, you digest and extract the RNA, which then is sequenced using standard bulk RNA-seq technique. But thanks to the barcode, you can then actually reposition these into the spatial context. So this is an extremely powerful technique, and there are a lot of progress there. And you can clearly see there are different clusters arranged by the software for healthy liver and PSC cirrhotic liver. Now, in order to understand whether there is a specific collagen signature into a spatial context, we have overlapped all different core collagens identifying PSC or in NASH ECM tissues, and score each of these spots within the tissue. So obviously, as expected, nothing new here, we were able to confirm that in NASH and PSC you have an increase of core collagen clusters, but then we were able to remap them remap them into the disease tissue. The other question is, okay, we know what this proteins are localized in the tissue, but which cells is actually contributing to the production of these specific matrixome components? So in this case, we have downloaded the single cell sequencing atlas from Ramachandran et al. And then using specific in silico tools, apply these into the spatial context. And here what you can see is that different cell types, actually not only fibroblasts, are involved in the production of the matrixome collagen signature. The limitation of this approach is that single cell sequencing studies are characterized by low number of cells, and also the definition of cell signature is not standard across multiple studies. So the last part of my talk, I will actually then move from the sort of biology to therapeutic target discovery. And in this case, it's something we started with Takeda several years ago in advanced NASH, and right now we have already started drug discovery for a few targets. However, using the collaboration and as part of the partnership, we were able to identify some pan-matrixome targets that we call matritarget X, and we then confirm biology, safety, trackability, human disease relevance, and so on, and also developed ECM-based assay to confirm the functional role of a certain targets into driving disease progression forward. So in this case, in order to develop the assay, we moved from the ECM scaffold, which is used for matrixome studies, so the intact ECM, into hydrogel. This allows us to retain the ECM biochemical signature from the original tissue. We lose, of course, the ECM treated architecture, but we are able to scale the format dramatically because we can perform basically high-throughput screening into micro droplets of tissue-specific diseases ECM, and including inside this hydrogel single or multiple cell type. So one of the key feature of this 3D ECM system is that it allows to mimic tissue fibrosis in vitro, not only by having secretion of procollagen or fibronectin and so on, but actually to have deposition into the tissue of ECM fibers, which of course is the key hallmark in tissue fibrosis. Using high-throughput spinning confocal techniques, we are also able, and our own algorithm, to quantify increased ECM deposition into the tissue. So by taking these pan-liver fibrosis matrixome targets and what we did was to develop cell-specific knockdown into the 3D ECM culture by knocking down this matrixome target X. Of course, for confidentiality, we cannot release the name of the target yet, into primary hepatic stellate cells resident into the system. So as you can clearly see into these ELISA graphs, is that after target knockdown, we were able to massively downregulate the secretion of CTGF, procollagen, and fibronectin, and also collagen 4. So this is basically secreted. In addition, we are able to induce a profibrogenic response and fibro deposition by adding a cocktail, profibrogenic cocktail of TGF-beta plus PDGF-beta, and after the target knockdown, we were also able to suppress fibronectin incorporation into the de novo ECM. So this target knockdown has the opportunity of suppressing collagen and ECM deposition in a fibrotic setting. We have also confirmed that this protein actually not only has a bioactive role in affecting stellate cell biology, but also has a structural role in promoting fiber formation. So here, just to see that the combination of collagen with the MATRI targets accelerates the fiber formation compared to the control. And lastly, obviously moving the idea of a target into a druggable targets. I'm not going to discuss for this lecture the therapeutics and drug discovery effort, but an important point is the possibility to measure these targets into the serum in order to have a target engagement biomarker, very relevant for drug discovery. And thanks to our colleagues at Takeda, we were able to confirm that the actually target of interest is elevated in patients with fibrosis and cirrhosis, and indeed also associated with advanced fibrosis. So to conclude, tissue engineering is a very exciting field. I fell in love with this 15 years ago when I was a student in Florence and then in London, has really multiple applications in the medical fields, including the possibility to discover some targets. Now for tissue engineering, you combine cells and scaffolds, and the best scaffold that we have in our hands is actually our own human scaffold that is called extracellular matrix. This is extremely bioactive, and we know it drives disease progression forward. In a specific indication of interest called PSC, we have confirmed that the biliary matrixome is characterized by a strong inflammatory and ECM remodeling signature. And by applying very advanced molecular techniques for deconvolving spatial and single cell omics, we've been able to confirm that it's not only fibroblasts that contribute to the biliary matrixome, although they are the most dominant cellular component, but also other cell types. And applying spatial transcriptomic into our pipeline for target discovery, we are able to remap into the spatial context the matrixome proteins into PSC tissue. Of course, much more opportunities for this. And of course, knockdown as a key aim of these research translational activities for therapeutic discovery, the possibility of knocking down a matrixome targets within cell types reseeded into ECM culture condition can reduce the stellate cell activation and the profibrogenic mediators and tissue fibrosis. And next, of course, I would say our personal goals and my dream since I started entering in love with biology and medicine is to develop novel therapeutics to treat PSC, fibrosis, and solid tumors. The most important slide is here. Obviously, this is a tremendous amount of work driven by an amazing group of people. So what I would like to acknowledge is the effort of this fantastic group of the team at Engitix, our friends and colleagues at UCL, Takeda, Royal Free Hospital, and Doppler Pharmaceutici. So I would like again to thank you all for the attention, and I look forward to receive your emails if you have any questions or comments, and to be there in person next year. Thank you very much.